Myself, Rupesh Undkar, studied at Ashok Mani Institute of Pharmaceutical Science and Research, Save. This video is guided by Mr. Azad Dhage sir. My topic is explaining two methods used for isolation of bacteria. Isolation of bacteria. Culturing is a procedure during microorganisms are cultivated in a culture media. Now, generally, when we culture microbes, we obtain a mixed culture that contains more than one microorganism. Pure culture is a type of culture medium that contains only one type of microorganism. Pure culture is usually derived from mixed culture by transforming a small sample into a newly prepared culture media. There are following methods by which we obtain a bacteria. First, strict plate method and second, spread plate method. First, strict plate method. It is the most widely used method for isolation of pure culture. Streaking is the process of spreading the microbial culture using an inoculating needle on the surface of new media. These are apparatus required for strict plate method are nichrome wire loop, Bunsen burner, nutrient agar plate, mixed culture of bacteria. Principle is different techniques used for purification of culture are strict plate method, pour plate method, spread plate method, and micromanipulator. Inoculation of microbial culture to the surface of sterile agar plate and spreading it by an inoculating wire loop is called strict plate method. Strict plate technique is used for cultivation, isolation, and separation of microorganisms from mixed population. Strict plate technique is used to produce well-separated colonies of bacteria from mixed suspension, and the size, shape, color, and other physical characteristics of isolated colonies are studied. One strict plate, two strict plate, and multiple strict plate are commonly used techniques for isolation of cultures. This is the figure of strict plate method. Procedure is prepare nutrient agar and sterilize by autoclave at 121 degrees Celsius for 15 minutes. Pour the medium into petri plates. Carefully rotate the petri plate. Take a small amount of bacterial inoculum by nichrome wire loop from agar slant or agar plate carefully. Lift the lid and streak the culture on the agar medium as shown in figure. Streak the inoculum and the flame the wire loop to destroy the culture present on the wire loop. Different techniques of streak may performed on the different petri plate using the same inoculum. Incubate the petri plate at 30 to 37 degrees Celsius for 24 to 48 hours. Observation is to observe individual colonies forming as per the streaking pattern changes. No contamination observed. Result: it found to be a successful strict plate method is well isolated individual colonies agar surface. The isolated colonies with no filter of contamination confirming that the strict plate method effective on pure culture. Second is spread plate method. It is the best method of isolate pure culture. In this technique, 1 ml of sample taken from diluted mixture which is then placed on the surface by using L-shaped glass rod called spreader. Apparatus is test tube, pipette, glass spreader, nutrient agar plates, Bunsen burner, and alcohol or acetone. Principle is the spread plate technique involves using a sterilized spreader with a smooth surface made of metal or glass to apply a small amount of bacterial suspended in a solution over a plate. The plate needs to be dry and at room temperature so that the agar can absorb the bacteria more readily. A successful spread plate will have a countable number of isolated bacterial colonies evenly distributed on the plate. This is the diagram of spread plate technique. Procedure: make a dilution series from a sample. Pipette out 0.1 ml from the appropriate desired dilution series onto the center of the surface of an agar plate. Dip the L-shaped glass spreader into alcohol. Flame the glass spreader over a Bunsen burner. Spread the sample evenly over the surface of agar using the sterilized glass spreader, carefully rotating the petri dish under at the same time. Incubate the plate at 37 degrees Celsius for 24 hours. Calculate the CFU value of the sample. Once you count the colonies, multiply by appropriate dilution factor to determine the number of CFU per ml in the original sample. Observation is a successful spread plate will have a countable number of isolated bacterial colonies evenly distributed on the plate. Calculation: if your spread plate is successful after incubation, you will get isolated countable colonies evenly spread across the surface of the agar. Count the number of colonies and multiply by it the appropriate dilution factor to determine the colony forming unit CFU present per ml in the original sample. CFU per ml is equal to number of colonies multiplied by dilution factor and divided by volume of culture plated. For example, suppose the plate of the 10 to 4 dilution yielded a count of 27 colonies, then the number of bacteria in 1 ml of the original sample can be calculated as follows. Bacteria per ml is equal to 27 multiplied by 104 multiplied by 10 gives 2.7 into 106. We have multiplied by 10 because we have used 0.1 ml while plating the agar plate. Result: a successful spread plate will have a countable number of isolated bacterial colonies evenly distributed on the plate.